Practical 1. Basic concepts of statistics and variation. The objectives in this practical are to calculate and describe the following descriptive statistics such as mean, variance, and standard deviation. Then, we need to understand the concept of correlations. Other than that, we also need to determine the relationship between the two variables. So, the materials that we are using are leaves, ruler, weighing balance, and graph paper. Let's proceed to the procedure. Firstly, 10 leaves were selected and their length and weight were measured. Next, the mean variance and standard deviation were calculated. This procedure was repeated for 3 sets of leaves, giving a total of 30 leaves. The combined data were used to plot a graph of leaf length versus leaf width. So next, let's take a look at the result that we have recorded.
Next, the graph that we obtain shows that leaf length changes with leaf width, but the relationship is not uniform. The data from distinct clusters indicating different leaf size characteristics, and suggesting that leaf width alone does not determine leaf length.
Now, let's shift our focus to our data analysis, starting with the mean measurements of the leaves. In this slide, we compare the mean length and width across the three different sets. While the lengths stayed fairly consistent, we notice more variation in the width of the leaves. This suggests that while the leaves are likely from the same species, environmental factors might affect their width more than their length. Next, we move to the statistical side of things, variance and standard deviation. Variance helps us see how much the leaf size spread out from the average. If the variance is high, the leaves are very different in size. We then use standard deviation to bring that measurement back into a simple unit, like centimeters, so we can see the average among the leaves differ from the mean. Finally, on the last question of practical 1, we address why it is necessary to have different sets of measurements. We don't just take one set because leaves vary naturally due to age and growth conditions. By using multiple sets, we increase our accuracy and make sure our final data is a fair representation of the entire population, not just a few specific leaves.
So, next, during our second lab, we have done the practical 2, which is titled Factors that Affect the Distribution and Abundance of Organisms. So, the objective in this practical are to introduce the simple concept of quadrat, to enhance the power of observation, to record the difference in plant distribution between the quadrat. Lastly, to infer from natural observations the possible factors that affect distribution and abundance of the organisms. And the materials that we are using here are raffia strings, measurement tape to ensure the length and width of the quadrat remain constant and the same, stick, scissor, and notebook. So here are the procedure for practical 2. Firstly, a quadrat measuring 5 meter to 5 meter was established and each forest type. Next, several quadrat samples were taken at random locations throughout the habitat. All three stands found within each quadrat were observed and recorded. The data collected were then used to estimate the population size and population density of the trees in the entire habitat. So here are the results that we have collected for practical 2.
Let's look at post-lab question 1. In our fieldwork, a quadrat is not just a frame, it's a specific ecological sampling unit. We use these square or rectangular areas to isolate and study organisms. By counting everything inside the quadrat, we can estimate the abundance and distribution of species across the entire habitat. Next, we move to question 2, which asks about the two basic requirements for this technique. For our data to be scientifically valid, we must ensure two things. First, random placement. We have to locate quadrats randomly to avoid sampling bias. Second, they must be non-overlapping. This keeps our samples independent and ensures they truly represent the whole population. Moving on to question 3, we look at the pros and cons of different quadrat sizes. If we use a larger quadrat, we get a better picture of species diversity, but it's very time consuming to count. On the other hand, smaller quadrats are much faster to process, but we might miss out on certain species if they are spread out in patches. Question 4 discusses the problems with spatial scale. Ecologists often struggle with large scale habitats like deep oceans or dense forests. In these cases, it's impossible to sample everything because the area is too big, accessibility is limited, and many organisms are simply hidden from view. Lastly, for question 5, we hypothesize why plant species are distributed differently. This is likely caused by variations in soil properties like nutrients and pH, as well as differences in light and microclimates. Biological factors like seed dispersal patterns and competition for resource also play a major role in these distribution patterns. That concludes our presentation. Thank you.